Hi, I'm sorry I couldn't be with you today, but I had an emergency at home and I had to leave school a little early. Very unusual, I know, but and I hope to somehow give you an opportunity to make a few bonus points right here at the end of the year to replace the points that we didn't get a chance to have fun with in a in-class quiz bowl. So let's talk first about what you need to do during this period. First, you need to do a course evaluation form for the main office and then another course evaluation from BioBook. What you're looking at here is the front cover of your textbook. And if we jump to the bulletin board, uh, you can actually see that there's links here. Your course evaluation forms are listed here. This is your NARICS course evaluation form. This particular one, if you click on it, it'll open the SurveyMonkey, and you need to select, the semester that you're in, my name on this list, and then the course, which is just biology. And then click on these buttons to indicate how you feel about these statements, and then maybe give a few comments about what you feel about class, and then click the done. Once you're done with that one, you need to do the BioBook course evaluation form. When you click on this link, a green screen comes up. You need to put your email ID on this one. This one, you actually get 10 points for filling it out. It is anonymous. If you read this up here, it talks about how the information you provide on the form is going to be good for me to actually improve the course. I hope to just take a look at all these things over break so that I can figure out maybe if I can do something that will help in the spring semester. Make changes at any rate. You'll notice the Likert scales here. Well, I'm trying the new thing on this drawing pen and it just went bonkers. Let's erase that and see if I can just try that again. So right here, this is the scale. 1 is poor, all the way up to 5 is excellent or very positive. You can't submit this form unless all fields are filled in. So this is kind of important right here. If you don't fill in all fields, it actually won't take the form. You need to put your email ID in without the @narinxhs.org. That I don't need. But the email ID allows me to keep track of who's actually submitted them and who hasn't. So for example, with the B period class, I've already got a bunch of submissions, but I have no idea who anybody is in the database. I don't. They're listed just by the order that people submitted the forms, so I have no idea who was first, who was last, and who submitted what. But without the email ID, it also won't submit the form. So basically, how this works is you scroll through the page, you click on the Likert scale buttons here, and at the bottom there's a few short essays, and then you click the submit button. And if things work, at the top of the page, right up here at the very top, there will be a little blue uh, notice right there at the top, right above survey form. It'll either say all answers were submitted or there was some kind of a problem. If you run into a problem, what you really want to do is immediately use your go back button on your browser and your answers will still be there. I have it coded so that would happen. And then you can click submit again and try another submission after you correct, hopefully, the problem. So that's your course evaluation forms. Be sure to turn those in today, or you can turn them in tomorrow. I'm sorry, today's the last day, so you need to turn them in today during class today. I'll check them at home and record your grades accordingly. Next, now we need to go to your um, main textbook again, your textbook front cover, and go to your first semester exam study sheet. So I'm clicking on 1S exam here, and here we go. So your exam study sheet is your study guide for the exam. If you don't use this study guide for the exam, you cannot expect a great score on the exam, okay? First, at the very top of the page, you have instructions on like just general things about the exam. And a couple things up here. Here we go again with the crazy. So at the very top, it says there's no bonus for finding mistakes on the page. This page is in a state of flux, and I am going to have mistakes on it. It's just I'm trying to get information to you as fast as I can. It was last updated yesterday at 8:22 p.m. So if I make any changes, I will change the date there. And if the date changes, you know that there's something new on the page. I usually put the changes in red. At any rate, if there's a mistake on the exam, please note the mistake on the exam. Remember now, I don't answer questions on test. I will not answer questions during the exam. If there are any issues on the exam, put your comments on the page where test comments belong. That'll probably be on a 5th page if I have to use 4 pages to give you plenty of room to write things down. If you think you needed clarification on a question or whatever, make sure you write that down. A couple things about math problems. Make sure you round all your math answers to the appropriate level of sig figs. I'm not a stickler on sig figs, but if you're writing down a ridiculous number of decimals, of course you're going to lose a little bit for being ludicrous on the exam. The exam is generally 4 sides. You might have a 5th side, which would be your comment question. I'm not done writing the exam, but I had to jump into this to talk to you so that I could communicate the basic exam instructions that the B period class got yesterday. If you missed the exam, you're going to be taking the makeup exam. That's right here linked. I have not finished that, but I will. Before you take that exam, you need to come and see me so I can talk to you about what is on your exam, and that way you can properly prepare for it. Otherwise, you're trying to study everything, and that is inappropriate at this time. So if you get sick or something, don't worry about it. Print the makeup sheet, which will be done probably by tomorrow evening, and then go ahead and see me. We might have to do a Skype if necessary, or I might have to send you a document with things circled so you know what's coming. At any rate, always do this on every exam page. Put your name in the name blank, the date in the date blank, and your class letter in the class blank. Please don't forget to do that. 2 points for the front page, 1 point for every page thereafter, and with a 4-page exam, you're looking at 5 points just for filling out the tops of the exam. For extended time students, you can tear off the front cover sheet, but be sure your name and stuff is on the other sheets. Keep the front cover sheet with the exam when you turn it in. Make sure you turn in all exam papers. If I don't get your front cover sheet, for example, and you're in extended time, you will get dock points for that, so make sure you turn everything in. There will be some bonus on the exam. I have no idea which question will be bonus. You can expect at least 1 or 2 questions to not be on the study sheet and on the exam, and those are likely your bonus. Now, I'm going to scroll on down to the exam breakdown right here, and then I'll jump up to pictures as necessary to talk about specific things. So, first off, your microscope will be on there. Now, there's 2 forms of the exam, and it's important that you remember that one exam might get the stage, the eyepiece, and objective, and the other exam might be the course in my fine focus and mechanical, stage or something. I don't know. At any rate, you have to know the basic parts that you use on a regular basis when you use the scope. So certainly the eyepiece, the objective, your focus knobs, the stage clip that's on the stage that holds the slide in place. All that stuff is pretty basic stuff, like the light bulb. You need to know that. So you will have the diagram, and the diagram you're looking at is this diagram right here. So this diagram is the one that you're looking at that's actually going to be on the exam. It's an exact replica. It's literally a picture of your microscope. Couldn't get more appropriate than that. So make sure you study it. That was what we started out the entire course with. Now jumping back down, the size of a microscopic object. We did this on a couple of different labs. So this question right here, I'm going to give you a little drawing circle, and you're going to have there's going to be a microbe in there, and you're going to have to use rulers that are already on the exam. So you'll have, see if I can work this out. So there'll be, let's say I go over here. There will be a drawing circle. You'll have a little microbe drawn there, and there will be a ruler provided here and here. And with the ruler, you can actually get the size of the microbe. If you want to get the size of the microbe here, fold the edge of the test over so you can use it as a ruler to get the measurements here. So if you're at a 2 here and you're at a 4 here, you have to take 2 minus 4, which is 2. And then in our drawing here, let's see if I can replicate this. Let's put this down here. All right, good. Now, let's get rid of that little blue section. All right, good. If you had 2 here, which you would stop doing that. And if you have a 4 here, your 2 minus 4 is 2, so you have to make sure you're using a difference here because your ruler will start with 0 at the edge. And then you remember now, you have to use your equation. You have to take this length divided by the width of the circle. That will be almost the entire length of the ruler, or just a little less than that. And you have to take it times the field diameter, and the field diameter is provided for you on the exam. So with that said, that's a couple of points on the exam, okay? So we did that a couple times. I think most of you know how to do this, but you want to make sure you review that. There are no pictures up here that match that part. Next part, cell matching. That would be this part. So I'm going to have this on the exam, and then I will ask you, you can certainly bet you're going to get the chloroplast or the mitochondrion or the Golgi apparatus or the nucleus or the rough ER. Now, one exam will get one group of those, and the other exam will get the other. I'm not likely to put the microtubules on. It's just I don't need that. You don't need that. But the vacuole is kind of big. That's something that might be on there, and certainly ribosomes. We talked about them in multiple chapters. That'll be on there. So you'll have to know both names and functions. So I might ask you, what's the powerhouse of the cell? And you have to match that to K right there. And then I might ask you, what's the name of that little organelle right there? And so I will say which organelle is a chloroplast, and you're going to say A. And then I'm going to say which organelle is the powerhouse of the cell, and you're going to say K. And then there'll be a fill in the blank, and it'll say which organelle stores water, and you're going to say C. Or I might ask you, hey, what's the function of organelle F right there? And that's to hold the chromosomes and protect them. So that's the cell structure, that's the cell diagram matching. That's the next part of the test. Now the cell membrane matching, that's going back to this diagram. I decided to use this, the sodium-potassium pump picture, because it's got the basic parts of the cell membrane here. So D and G and H, those are proteins. So this is a protein, that's a protein, and that's a protein, okay? Elsewhere, like these other pointers over here, like A, B, and C. A is a phospholipid, and B is a fatty acid, and C is a phosphate. So these are not pointing to proteins. These are pointing parts of phospholipids. Now, you got F is the lovely ATP molecule here, which is providing energy to G so it can do the pumping. And E, which is this part right there, it's got those little balls right there. Though those are sodium, remember, ions that are going to get pumped out. And these are potassium ions that are going to get pumped in to the cell. So one more time, D and G and H are proteins. G happens to be an active transport protein, while H and D are like channel proteins, gates that allow chemicals to flow through. But D and H don't use any energy, and G does use energy. All right, let's go to the next part of the test. That is talking about scientists. Now on this is a big matching, but you know, these are important people. They're part of the reason we're all alive today. These folks, some of them literally invented cell biology. Some of them were important to chemical activity inside of cells. Without their contributions, we're still shaking gourds and going hiya hoa when we get sick. So these folks are really important, and you can bet you're going to get like Daniel Arnon, for example. I know he was very important over here, but you know, probably he's not on there. But you can bet like you're going to get Rosalind Franklin for sure, because it's kind of important as a important woman in science. She made such a massive contribution and was totally underappreciated. And I like the underdogs, so Rosalind's definitely there. So you'll get probably 4 or 5 scientists out of the list. I don't know which ones are going to be in your matching section, but look back at your bioassigns, look back at your tests, and you will see that there are specific statements about the scientists, and those are the ones I'm most likely to pull and put on your exam. Remember, there are 2 forms of the exam, and sometimes it's difficult to come up with 2 different statements for each scientist that's going to be different for each test. So some tests might have different scientists to make sure that the tests are different. Next, to fill in the blanks, I don't know if we're going to have any of those, but if we do, it would be probably no more than 4. And if we did have those, I would probably try to narrow the field. But right now, I have not finished making the exam, so I really don't know exactly uh, which ones you're going to have. Next, there definitely will be an essay question about osmosis or active transport on your test. We spent a good day talking about osmosis. It's extremely important. I just had a student tell me or ask me a question tonight. Hey, Mr. Kensting, what would happen if you died from drinking water? If you died from drinking water, it could be that the water had toxins or pathogens in it and they made you sick and you died. If you died from drinking water, it could be because you drank so much water, you literally overloaded your digestive tract, excess osmosis occurred, and you died from diluting the sodium in your blood. And that's an osmotic thing. And you could die from drinking water if the water actually went down your windpipe, and then you would choke and or drown at the same time. Osmosis is a big deal. Make sure you know about osmosis and make sure you know about active transport. I spent a good part of the class talking about the sodium potassium pump. So make sure you look those two over because they are definitely going to be on your exam. Not real big on the facilitated diffusion, but it's there just to remind you that that's something that happens. And I should mention up here on this picture, this is a facilitated diffusion protein. This is a facilitated diffusion protein. This is an active transport protein. And none of these three deal with osmosis, but water molecules shooting through, like right through there, just shooting through from one side to the other, that's osmosis, okay? All right, so let's scroll back down here and look at these next parts. You will get an osmosis question. Oh, wait, let's go to the next one now. Biochemistry matching. Biochemistry matching is talking about these biochemicals. I can pretty much guarantee you're going to have, whoops, let me get rid of that. This is on there for sure. I mean, that's glucose. It's absolutely famous. If you don't know what that is, you got to get it in your head for the exam or you're going to get that wrong. That's a big one. This is another big one. That's a little amino acid. You ought to know that. It's also called a monopeptide. If you don't know what that is, that's another problem on your exam. This is another biggie. That's what makes our food so tasty. It's in ice cream. It's in butter. That's a triglyceride. And because that's such a ubiquitous part of the American diet, you really need to know that one. Probably, you know what? The dipeptide and the disaccharide up here. So this one's a disaccharide. This one here is a dipeptide. Those two may be on the exam. I mean, you're probably going to have 4 or 5 total chemicals, and you'll have to do some like matching, like which one is glucose, which one is disaccharide. I can tell you this one down here is a polysaccharide right there. So you might get a question that says which one could be starch because that's a polysaccharide. Not likely on the exam, but I leave them here as in case I gotta have them because I have to make 2 sections, 2 forms of the exam. You have DNA here. This is DNA. It's double-stranded. You can see the little nucleotides, nitrogen base, sugar, phosphate there. And that's the little L-shaped structure right there that we were talking about. That's a nucleotide. All right, and then this right up here, this is a steroid. That is the signature shape of a steroid. They have that goofy hexagon, 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 pentagon shape. That's like testosterone, estrogen, cortisol, cholesterol. They all have that formula. So you're going to get questions like, which one of these is used by ribosomes to build a protein? Well, if you had that question, that would be this guy right here. That's an amino acid. Okay. If you had which one is produced by plants by photosynthesis, that would be this one. That's glucose. Okay. If you had a question that said, which one makes butter the tasty food that it is? That's this one, which is a triglyceride. Okay. So you might also just have a question, which one is a dipeptide? That would be this one. That's the dipeptide. Okay. Now, that's it for the chemical matching. I know a lot of you just really love the biochemistry part, so that part is right up your alley. The next part is, you know what? This is a double question here. I need to get rid of it. You will definitely have an osmosis question on your test. You might be in a swimming pool that's freshwater, or you might be in a saltwater swimming pool. Be ready to talk about what happens to the human body in those situations. Now, the photosynthesis and respiration diagram, I can't get both of these diagrams on your test. They take up too much room, and I don't want to use that much paper. So I pretty much I'm gonna put one of these on one test and one of them on the other. So I have no idea which one you're going to get. Make sure you know both of them, and they were that foundational to the chapter. It's not like there should be a surprise at this point. You can bet that for those 4 pictures, you're going to have to say things like, you're going to have to know where glucose, or can I not make you blue? Okay, let's click. Okay, it's not wanting me to unclick. Okay, there we go. You can bet you'll have to know, like, where is glucose or carbon dioxide or oxygen or water made or produced. So let's go over that really fast, because those are the major players of cellular respiration and photosynthesis. So on the diagram on the left, glucose, if I can get this thing to work correctly, that right there, this is glucose, and that's being made by the dark reaction right there. Carbon dioxide is being consumed by the dark reaction right there. That's carbon dioxide going in, so you can see an arrow, it's coming into the dark reaction, going that way towards number 8. I can also tell you that right there, number 3 is water. Okay, why it made a giant spike, I don't know, but that number 3 right there is water. Water is an important component of photosynthesis, because it's split to release electrons that come down this ETS right through here, this electron transport chain that's powered by the chlorophylls at 12 and 13. They're called photosystems. But 4 is oxygen gas that's released as a waste product from breaking water down in photosynthesis. So what we have here then, the 4 major players, one more time, we've got glucose, carbon dioxide, water, and oxygen. Okay. Because I got to make 2 exams, this is the ATP shuttle and this is the hydrogen shuttle. Probably leave most of the other stuff alone unless I ask you, what reaction is this? This is the dark reaction. This is the light reaction over here too. Now let's go over here to the reactions on the right. This is respiration. So let's talk about those 4 chemicals now over here. Over on the diagram on the right, glucose is right here at 4. So there's glucose right there at 4. Let's get this. Nope, I just accidentally launched VLC. Okay, we don't want that on the computer here. So hopefully it stays in the background. So we've got 10 right there. That's glucose. I'm sorry. No, not glucose. Etch that. Number 4 is glucose right there going in. And carbon dioxide is coming out at number 11 right there. So you can see that glucose is consumed by glycolysis at 1. And carbon dioxide is coming out of the Krebs cycle at 2. This is the Krebs. And then the oxygen gas that we breathe in is being consumed at number 6 right there. Number 6 is oxygen right there. And when oxygen is consumed, it combines with electrons and hydrogen coming off this ETS here at 3 to become water at 7. Now, there's a lot of other chemicals on here, but you know what? We just can't ask everything. There's only 2 last things that are significant here: hydrogen and ATP, which was significant in photosynthesis too. So ATP are being made at 10 right there. And hydrogen is at number 8. It's being kicked out by the Krebs cycle all the way around. Let's get there we go. And hydrogen is also being kicked out over here in glycolysis, and that's what's going to get picked up here and sent to the ETS down here. So again, one more time, 4 is glucose going into glycolysis. And then the products of glycolysis, you remember, was pyruvate, but that's probably not going to be on the exam. That goes into the mitochondrion, and then the Krebs cycle smashes up the pyruvate and cranks out carbon dioxide waste. The hydrogen atoms that are left over from that smash up go to the ETS, blah, 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 they go down. And then they combine with oxygen gas to form water at 7. So oxygen 6 and water 7. Okay, the CO2 coming out's at 11. And then lastly, ATP is being made here at 10. And the other precious commodity here is the hydrogen atoms, which are at number 8 coming off of Krebs. So that's kind of your exam is kind of a summary there. You don't have to know all the specifics of the reactions. And your reactions will be one or the other. I just don't think I can fit both of them on the exam. It's just going to take too much space. Next, DNA activity matching. That's down here. So your DNA activity matching, you're going to get, I had to choose one of the diagrams, and I decided that since this diagram was more generalized, you're going to get this. So you need to know that A is the location of transcription and B is the location of translation. That's the first thing you for sure got to know. And then you got to know what these chemicals are out here. So I might ask you, what happens at A? And you say, well, that's transcription. I might ask you what happens at B, and that's translation. At A here, you have the production of messenger RNA. That's where this thing came from right here. This thing was made in here, and it slipped out of the nucleus, and it's going to crawl down to a ribosome. So this is in here is DNA, and it transcribes RNA, and that's called transcription. At B, we have translation. This is a ribosome. It will use this little messenger RNA here, and transfer RNAs will bring amino acids, and proteins will get made. They're called polypeptides technically. These little guys are amino acids at 4, and they're going to be condensed into a protein by the ribosome when the transfer RNAs bring them to the ribosome. So make sure you know this diagram right here. It's basic DNA activity in a cell. That's kind of important. The replication bubble. The replication bubble is this. I need to ask you a question about replication. It's one of the most recent topics we did. So at any rate, this diagram right here is definitely on the exam. And you might have the 5' on the upper left. It might be on the lower left. Please do not memorize that the leading is always, right there, because you may end up getting it wrong. So on this particular diagram, from here down is leading, from here over is leading, and then lagging, and it's hard for me to draw the arrows with this stylus, are over on the right, and this is lagging down here too. So I might ask you, what's label the helicase areas, which are there and there, and label the origins, which are here and there. At any rate, so you will have a bubble thing on there. And then lastly, well, actually not lastly, I got a couple last things to say. There is a scientific analysis question. I haven't decided whether it's going to be the yeast fermentation lab or the enzyme lab. They both had pretty simple data to work with, so you're going to get one of those two, and one test might have one, the other test might have the other, and you're going to be needing to graph the data, and then you'll have to, title and label your graph, and then you'll have to draw a trend line. Then you'll have to write down the coordinates of a point, okay? So make sure you review how to do that. Then you'll have to calculate the slope of the line, okay? That's important. Make sure you do that. We've been doing that on numbers of tests. And then you'll have to report the slope with coordinates, okay? And don't forget your coordinates. And make sure you reduce your fractions. Do not write 2/7 as the slope. Use a calculator. You can have one on the exam, but it can't be a programmable one. You can divide the 2 by the 7 and get a decimal. It will be a decimal, right? Record the decimal, round it to sig figs, and then put the units after it. And then you'll have to tell me what the meaning of the slope is, which is always how some y value changed with respect to one x value. So if you were measuring raindrops per minute outside in the courtyard, if your slope was, let's say your slope was 5, and it's raining really slow, 5 raindrops per minute. I can't really write too well with the stylus, but if it was 5 raindrops per minute, and your thing was be RD per minute, that looks kind of horrendous, then you would report the meaning of this as it rained in the courtyard 5 drops, DROPSPERMINUTE. Do not write the symbols out. You have to write the words out to actually get credit for the problem. Now, there's a few pictures up here I didn't talk about, and they're here. I will guarantee, and I haven't added this part yet, you will definitely get the enzyme thing. That's kind of a big deal. It was on your test. It's definitely going to be on the exam. So you're going to need to know what these parts are. One is pointing to this part right here. This is the enzyme right here. Okay, that's the enzyme. 2 is pointing to the substrate. Okay, substrates are things that enzymes attach to. 3 is pointing to your induced fit model of enzyme activity. It's where the substrate literally binds to the enzyme at the active site, and the two literally meld into each other. At that point, then a change occurs, and you get, notice this is hydrolysis. We have a dimer becoming 2 separate monomers. There's no bond between them. So 4 are products, okay, which happen to be monomers of this reaction. 2 is a reactant, and it's also a substrate, because in this reaction an enzyme is used. If there was no enzyme involved, this would just be a reactant, and these would be products. But since this enzyme is going to break this thing down, this is not only a reactant here, it's also a substrate to this enzyme. So here's the induced fit part, and then this is the active site at 6 of the enzyme that binds to the substrate, and 4 is the products. And where 5 went, I don't know, but if I add it, I will alert you that you need to look at this. The last thing, we kind of made a big deal out of this, hydrolysis and condensation, and that reaction right there summarizes both. So the reaction to the right, this is a polymer, is reacting with an enzyme, but you don't see the enzyme in this reaction. It's becoming a monomer and a dimer. In other words, it's kind of like what you see underneath it here, except here we're just not showing the enzyme. We're just showing the chemicals. This is a reactant, and this is a product, and that's a product. This happens to be a polymer. This happens to be a monomer, and that happens to be a dimer. And the reaction to the right, the one going to the right, is hydrolysis, okay? Because we're breaking a bond and making things turn into little pieces. On the other hand, okay, make the blue go away, computer. Okay, thank you. The other reaction is to the left, and in this case, we're taking a dimer, we're adding it to a monomer, and an enzyme puts these together and turns them into a trimer. This is a trimer or a polymer. That reaction is called condensation. So the reaction to the left is called condensation. The reaction to the right is called hydrolysis. There certainly will be some kind of question about that on the exam. Now remember, if I add anything to the exam, what I'll do is I will put a note on the study sheet here. I generally put it in red like you see on number 13 here. And then up here I'll change the update. This won't say 12:12 anymore. It'll say like 12:13 or 12:14. Now I'm not going to change anything tonight, but it could say 12:14, and then it will give you the time it was changed. Ladies, I'm not trying to trick you or anything. The only reason I'm going to put updates up is to alert you to the exam questions. I have not fully made the exam. I have been very busy. I got your quiz bowl together and stuff, and I just haven't had time to get all the specific questions on your exam yet. I'll probably not add any change. I won't add anything to this study sheet on Sunday. But the thing is, I could leave the study sheet as it is right now. But I can tell you this, your exam may have questions on it that aren't on here. The exam has to be made first before the study sheet gets made. So I know I've already experienced a little bit of frustration with the study sheet. Bottom line is, teachers, don't assume your teachers have to make study sheets. We don't have to do them. In college, you generally don't get study sheets. But I want to get you ready for the exam. I'm doing the best I can. I'll try not to put any changes on the study sheet, but if I cut it off right now, I will guarantee there will be questions on the exam that are not here because the exam isn't made. So if you have questions now, ladies, you got to email me. And so I can answer them. I will be at school on Friday, tomorrow, but I will not be there probably past 1 o'clock. Of course, I don't know. If the crew doesn't show up at my house that I have to meet, then I get to stay at school. Personally, I hate missing school in general, and I certainly hate missing school on the last day. But I can't do anything about this. I'm caught in a bind. If I can be at school, I will be there, and you will not have to listen to this, although I will provide this link as a backup anyway. So again, email me with questions. Don't forget to look at do your course evaluation forms, both the NARICS one and the other one, which is the BioBook one, which are on Canvas. They are on the bulletin board, and they'll be also in an email provided to you. Have a good evening. I hope you have a good weekend. Good luck on all of your exams. You're going to rock the bio exam.